Hello, and welcome to our Plant Evolution Trail. I'm Beverly Glover, I'm Director of the Botanic Garden here in Cambridge, and this trail is going to be presented by me and by Hamish, who's one of my PhD students. And what we're going to do today is take you on a walk through 470 million years or so of plant evolution, starting with the green algae, which are the closest living relatives of those early ancestors and those early plants that made the transition onto land. And then we'll show you some of the plants that are alive today that are representative of the different groups of plants that then evolved. We'll start with plants that have very poor waterproofing, very little ability to transport water around their bodies and have to stay very small and live in damp places. We'll then move on with the evolution of plumbing systems in plants to plants that could get taller and could grow in drier habitats and put roots down into the soil. And eventually we'll move through the plant phylogenetic tree into the seed plants, which are able to live all across the surface of the earth in a whole range of different habitats, and show you some of the great living representatives of those groups as well. The trail was designed to support the first year course in evolution and behaviour at the University of Cambridge, but we hope that it'll be interesting for people studying other courses as well, and that you might learn something about plants and enjoy it, whatever your background and whatever your interest. Thank you for joining us today.
Before we go into the trail video, there's one more thing we need to talk about, and that's the phylogenetic tree which we use in this trail. Now, a phylogenetic tree is a diagram which shows the hypothesized relationship between organisms or groups of organisms. So in this case, we're going to show you the phylogenetic tree of the land plants, with the liverworts at the base, then the hornworts, then the mosses, then the vascular plants going. But there's a problem. Phylogenetic trees are based on data, and they're based on statistical treatments of that data. So they're only hypotheses. They're what we think is the most likely explanation for the evolution of these different organisms. We can't ever know what actually happened in the history of evolution because we weren't there. And the problem is with the bryophytes. And the bryophytes are liverworts, hornworts, and mosses. They grow in damp cracks like the ones between these bricks here. And in the video, we're showing you the liverworts, hornworts, and mosses at the base, and then the vascular plants further up. And the support for that is morphological. Liverworts don't have a very good cuticle, they don't have stomata, they're really not especially good land plants at all. And then the hornworts and mosses do have a much better cuticle. They have some stomata, they're not very good, and there aren't very many of them, but they are still there. So the morphological data supports the phylogenetic tree, which we'll show you in the video. But the problem comes when we start looking at genetic data. We've got lots more genetic data over the last couple of decades, and that is showing more and more conclusively that the phylogenetic tree we're showing you in this video is wrong in the context of the bryophytes. What it's showing us is that the bryophytes are actually a monophyletic clade. All of the bryophytes evolved from a single common ancestor. And so in the lectures which Professor Glover is giving to the first year evolution and behaviour students this year, she's going to show the bryophytes as being a monophyletic group, with the hornworts sister to the liverworts and mosses in it, and the bryophytes being sister to all of the rest of the vascular plants. But hang on, I hear you cry. What about stomata? And this is the big problem with this new version of the tree. What it means is that stomata evolved at the base of the land plant lineage when plants moved on to land, and then, at some point in history, the liverworts lost the stomata. Hornworts and mosses still have it, the liverworts lost it, and the vascular plants, obviously, they still have stomata as well. I've talked to Professor Glover about this. We don't like it very much. We don't like the idea that liverworts lost these stomata and use air pores instead. However, the genetic data is so compelling that this is now what we feel is the most likely explanation for this. It may be in a few years' time you come back and we have a new introduction to the video explaining something different. Because remember, phylogenetic trees are only hypotheses. But for now, this is our best guess as to what's going on. So with that in mind, I hope you enjoy the trail.
Before we head into the glasshouse to have a look at some actual plants, there's a really important concept which we need to introduce, which is the alternation of generations. Animals, like you and me, are diploid. We have 2 copies of each chromosome in each of our DNA-containing cells. And our sperm and our eggs are produced by meiosis. They're haploid, they have only one copy of each chromosome, and they fuse to form a diploid embryo, which goes on to form a diploid individual. And that diploid individual will eventually produce sperm or eggs of its own. In plants, the first part of that process is exactly the same. The sperm and the egg will fuse to form a diploid embryo, which goes on to form a diploid individual. But instead of producing sperm or eggs, that individual will produce spores, and those spores are haploid. They have only one copy of each chromosome. The spore then germinates into an individual which is also haploid, and that haploid individual then goes on itself to produce either sperm or eggs. The sperm or eggs will then fuse to form a diploid embryo, which will develop into a diploid individual, and that whole process starts again. Now, the alternation of generations refers to the alternation between the haploid generation and the diploid generation. Now, most of the plants that you'll see around you, trees, flowers, conifers, they're all sporophyte dominant. All of the structures that you see here are diploid. They have two copies of each of the chromosomes. If you want to see a gametophyte dominant plant, you'll need to go to the bryophytes, to the liverworts, hornworts and mosses. They're all gametophyte dominant. The structures that you see there will have one copy of each chromosome per cell. But hang on, I hear you cry. I know that plants, flowering plants, produce eggs and sperm wrapped up in pollen. But I thought you said that the things that we see here are sporophyte dominant, so surely they should be producing spores. And that's a very good point. And what's happened here is that the sporophyte generation does produce those spores which will germinate into a gametophyte, but that whole process takes place within the body of the sporophyte. So within the developing anther or the developing ovary, the entire gametophyte generation is contained within there. It only lasts for a few cell divisions, and then they will produce the sperm, which is then wrapped up into pollen grains, or the eggs, which are found in the ovaries, which can then take part in the process of pollination. We'll see more about all the plants through the family tree as we go, but for now let's head into the glasshouse and have a look at Chara and Alga right back at the base of the plant family tree.
So I'm here in the Life Before Flowers glasshouse at the Botanic Garden. You can see I'm surrounded by fantastic plants that are all predate the angiosperms. These are all plants that evolved before the flowering plants. So we're at the start of our plant family tree here, and I've come in here particularly to show you the plant in the glass pot down here next to me. So tucked away at the back in here we have Chara vulgaris, the common stonewort. And it's not really a plant. If you look at the phylogenetic tree, you'll see that really it's a green alga. It's one of that group of green algae which contain the common ancestor of the green algae and the land plants. So Chara here, the stonewort, doesn't look very much. It lives in the water. It doesn't need waterproofing. It doesn't need the same sorts of mechanisms for support and gas exchange that land plants need, but it does keep its eggs attached to its body to give them protection, and that's the start of the land plant lineage. That adaptation was really important when the plants moved onto land.
Chara actually needs water to survive. It needs to be inside the water. But we're going to look now at the bryophytes, which are some of the earliest plants to make the transition onto land. And these are the liverworts, the hornworts, and the mosses. And I'm here next to one of the liverworts. This one's Conchocephalum conicum, the great scented liverwort. And it's got this thalloid structure. It's sort of small and squelchy. It's not differentiated into leaves and stems and anything like that. It just sort of spreads out. It's got some waterproofing, and the waterproofing isn't so much to keep the water off, like we wear waterproofs. It's more so that to stop the plant drying out. It doesn't have roots. It's got rhizoids, which are sort of root-like structures which help anchor it, but they don't take up water either, so it just needs to be in contact with the soil. Now this is the gametophyte stage of it, so it's haploid, and it will put up these little structures. I've got a picture here of Marchantia, which is one of the liverworts that we use in the lab, and this one here has got male structures on it. You get male and female plants, and there's different ones for the female. They look like spiders on the top of stems. And they need water for the sperm to be able to swim to the egg, And then when they fertilise, they will make a sporophyte which stays within that little umbrella structure and eventually generates spores which get released off to go and make a new sporophyte liverwort somewhere else. We've also got the hornworts. Again, these are small and squelchy things. The sporophytes within the hornwort are the horns which give it its name. They come up and they will release the spores from there. And finally, in the bryophytes, over here, we have a moss. This one is Polytrichastrum formosum. Now, mosses are a little bit more three-dimensional than the liverworts. They've got these leaf-like structures arranged in whorls around sort of stems, but they don't really have any terribly good plumbing. So again, they need to be based around wet soil. Now here I've got one which I found on my shed roof, and you can see the sporophytes. So again, the sperm and the egg fuse together within the moss, and then the sporophyte will grow out of the moss. It can't photosynthesize, so it's parasitic on the gametophyte, and it comes up with this little stalk-like thing which takes 3 to 6 months to grow. It's quite slow growing, and eventually it will release those spores out for more mosses to be able to grow.
So the plants we've met so far have all been gametophyte dominant. They spend most of their life cycle in that haploid stage where they only have one copy of each chromosome in each cell. The plants we're going to meet next, though, are in the lycophyte group. These are further along the plant evolutionary tree, and these plants spend most of their life cycle in the sporophyte stage, the stage where they have 2 copies of each chromosome in each cell. Now, the common name of these plants is misleading, so they're often called the club mosses or the quillworts. And I don't think any of those are very good names. They're not mosses, and I would prefer to just actually use lycophytes, which is, I think, no more confusing than any of the so-called common names. So the first lycophyte I want to show you is here in the tank with Chara that we met earlier, and this is Izoetes. Common name is the quillwort, and it's the little plant here at the front with these quill-shaped leaves. So it's a plant that lives in stream beds, in wet habitats, underwater, but it is a full lycophyte. It is sporophyte dominant, and it has spores produced on those on sacs that are grown on those leaves. So here's Selaginella, another one of the lycophytes, and it has these lovely little leaves that we call microphyll leaves. They're very small, and they evolved independently from the leaves of more common everyday plants like the flowering plants and conifers. And you can tell that because these tiny leaves only have a single leaf vein in them. They never evolve the ability to branch those veins, and so they can't get very big because the cells all have to be within a certain distance of those veins. So we've got several examples here behind me of Hupertia, which is one of the club mosses, another one of those groups within the lycophytes. And you can see that they're trailing plants. You can see where the name club moss comes from because they look quite moss-like. But again, they're sporophyte dominant. They spend their life in the diploid stage, produce spores, and then the gametophyte is very small and reduced. So now we've seen all of the plants that we want to look at in the Life Before Flowers glasshouse, we're going to go on outside. The plants we've seen so far are all small. They need damp conditions because they don't have very good waterproofing. They have to be in this warm, protected glasshouse. But the evolution of better vascular and support systems meant plants could get bigger and start to grow in drier environments. So let's go and have a look at some of those.
This is a tree fern. It's called Dicksonia. It's native to Australia, and it's not really a tree. This is not a trunk. This is the remnants of all of these fronds which have grown up. We actually cut them off to make it have a nice looking trunk to support it. But in the wild, they would simply fall off. And the fronds grow up right the way down from the bottom. They go all the way up through here and produce these beautiful structures. Now, these can be some of the largest leaf-like structures in the plant kingdom. They can grow up to about 4 meters long. They're absolutely huge. And they look very similar to some of the earliest fossils that we can find, fossils of the earliest seed plants that we can find. So it's likely that the earliest seed plants would have looked very similar to these. They have plumbing. They have systems to pull water up from their roots right the way up into these fronds. And this is the sporophyte. You will find underneath the leaves, I haven't got any on this one, but there's a photo here from a different plant which shows them. These sori, these little spots of the sporangia, which release the spores, which drift off and eventually germinate into a gametophyte. The gametophyte is tiny, it's only a few hundred cells, and the sperm will swim to the egg, and from that fertilized egg a new sporophyte will grow. So I've come back inside the Life Before Flowers glasshouse to show you a close relative of the ferns, which are the horsetails, and they're these rather spiky things here. They're relatives of the ones which you'll find in gardens. You'll know if you're a gardener or your parents are gardeners that the horsetails you get in gardens are almost impossible to get rid of. They spread viciously with underground suckers, and the roots of these ones go down about 1.5 meters, and they can regenerate from even a tiny little bit of root. So that's why they're so hard to get rid of. They feel very rough. You can probably feel the roughness of it on here. And that's because they've got silica in the cell walls. The leaves have degenerated to tiny little sort of runty little structures on the sides here. Now these are, again, the sporophytes, and unlike the ferns where you've got those little spots of sporangia, these ones bear their sporangia on the tip of one of these structures. And again, the spores will disappear off into the wind. These spores are actually fabulous. They're small, but they've got 4 little legs on it, a bit like a spider with only 4 legs, if you like. And in the wet, they will coil up, but when they're dry, they stick out. And with the cycles of wetting and drying, these spores can actually move, they can walk, and they can also jump, given the right conditions. And spores, they're absolutely tiny, but they can jump up to about 1 centimetre, which is an enormous height compared to the size of that spore. And that's enough for it to catch the breeze and be carried off by the wind. Also in the Life Before Flowers glasshouse is the whisk fern, Psilotum nudum. Now these don't produce leaves at all. All of this is stem, and it's all photosynthetic. The little yellow balls that you can see are the sporangia, and that's what release the spores. The whisk fern is epiphytic, so it doesn't have any roots. It relies on moisture and dirt from its surroundings to be able to get all of the nutrients it needs.
So all the plants we've seen so far depend on water for their reproduction. Their spores might be waterproof and are great for dispersal, but when they come to actually sexual reproduction, their gametes have to be accessible to each other. The sperm have to swim through a water film to be able to reach the egg and fertilise it. Now that's fine, but it does limit the sorts of habitats you can grow in, and also your ability to respond to changes in climate and even season. Now all of that changed with the evolution of seeds. The seed plants have solved this problem of reproduction on land by a very clever trick of keeping their spores attached to the parent plants. So particularly on the female side of things, the spore is retained by the sporophyte generation. It's not released. It germinates into the gametophyte, and then the gamete itself, the egg, all retained on or in the parent body. And that means it's protected. The sperm is wrapped up in a pollen grain in a waterproof coating, and it travels through the wind or through an animal to find the female parent, and then the sperm is able to fertilise the egg. And so the embryo that's produced from that fertilisation is again starting life protected by the parent and can be wrapped up in various sorts of waterproof coating. So this whole change in the reproductive strategy allowed plants to move into other habitats and to live throughout different seasons and to cope as climates changed. Now I'm standing by one of the cycads, and they're one of the earliest evolving groups of seed plants. So this is the cycad here, Dioion, and it's a representative of a group of plants that were very common about 200 million years ago, although now there are only about 100 species left. They have these big dissected leathery leaves, and these are thought actually to have evolved as an adaptation to avoid herbivory from the big herbivorous dinosaurs that were present at the time that the cycads were common. It reproduces just as I've described, and this particular plant gets its name because it produces 2 eggs, hence Dioion. Those eggs in pairs are retained on the parent plant and are fertilised by sperm that travels to it. Another group of the gymnosperms is the ginkgophytes, and they're represented these days by only a single living species. This is Ginkgo biloba, our first tree. And ginkgo is representative of a group that was very species rich, very common about 300 million years ago, but has died out apart from this one single species. And even this was thought extinct in the wild, but it was preserved in Chinese monasteries and temple gardens and is now quite widely grown in cultivation. It's an unusual tree if we compare the leaves to our more common broad-leafed plants, because you see that instead of a single midrib, a central midrib in the leaf with veins radiating out from it, we have a fan of veins. So it's a very different leaf structure, and that's because it's an example of convergent evolution of the broad-leafed form. We've met our first tree with the ginkgo, but I'm standing now by a representative of the conifers, a much more species-rich group of gymnosperm trees. This is Sequoiadendron giganteum, giant redwood, and it's an example of the biggest living organism that's ever lived on the planet. They can also live for several thousand years in the wild, so it's also one of the longest lived organisms we've ever had. It's a fantastic tree. It's got this amazing spongy bark that provides protection from fire at the base. It's got wonderful hanging foliage, and it produces by cones. It reproduces by cones at the tips of those branches, and those cones are designed such that they open after fire, and that releases the seeds when there's been fire, when there's clearance of the ground, to allow new saplings to grow and develop. There are about 700 species of conifer. It's not that many when you think about it, and particularly when you think about how dominant this group of plants is, particularly in the northern hemisphere, where they occupy great swathes of land in Canada, North America, and across Russia as well. But it's a particularly fantastic example to come and see the giant redwood, one of my favourites. So, throughout this trail, Hamish and I have been alternating plant groups, but I've got two in a row here because the next group we want to tell you about are one of my favourites, and that's the Gnetophytes. Now, the Gnetophytes are quite a small group. There are only about 70 species across 3 genera, and we're lucky enough to have all 3 of those genera represented here in the Botanic Garden. So I'm sitting next to the first one I want to introduce you to. This is Welwitschia, and Welwitschia is a very unusual plant. It actually has an underground trunk and these very long strap-shaped leaves. They grow from the centre, they abrade away at the end on the sand, and a plant can live for decades, and they're found only in the deserts of Africa. So Welwitschia is not a flowering plant, not an angiosperm. It's in the gymnosperm group, and as such it reproduces by cones which are produced from the centre here. The other Gnetophytes reproduce in the same way, and that reproductive strategy combined with the strange leaves led people to wonder quite what their position was in the plant family tree. We're going to show you Gnetum next, and Gnetum is probably my favourite plant of all in the Botanic Garden, and it's the genus for which the Gnetophytes were named. So this is Gnetum nemon. It's a lovely tree, rainforest tree from Malaysia, and you can see that it has beautiful leaves that look just like the leaves of a flowering plant. They have a central midrib, they have sensible veins branching off them, but if you look at its reproductive structures here, you can see that they're much more like small cones, the sort of cones you would see on a fir tree, than they are like the flowers of an angiosperm. The final genus in the Gnetophytes is Ephedra, and we've got quite a lot of different Ephedra species growing outside at the Botanic Garden. They make these large, almost broom-like shrubs, but if you look closely you can see that there's no leaves at all. It's the stems that are photosynthetic, and they produce these strange fruiting bodies that on a quick glance look like berries, but close up again are cone-like structures with this fleshy texture around them to attract birds to disperse them. So the interesting thing about the Gnetophytes is their position in the family tree. Because they have this combination of an angiosperm-like leaf structure in some cases, but conifer-like fruiting structures or cones, they were thought of as a sort of missing link between the flowering plants and the gymnosperms. But once we started using DNA to analyse relationships, it became clear that they were no such thing. They are fully embedded in the gymnosperms, and the phylogenetic analyses using DNA either embed them actually within the conifers or as a sister group to the conifers within the gymnosperms. So they're essentially conifers, fir trees, that independently figured out how to make leaves like the leaves of Gnetum, attract birds to disperse their fruit like you see in Ephedra, and came up with all sorts of clever strategies to survive in some very strange habitats.
Arguably one of the greatest drivers of diversity within the plant kingdom is the presence of flowers, and we're going to start looking at the angiosperms now. The angiosperms encompass the vast majority of plants. It's probably about 90% of plants are flowering plants. It's about 300,000, 350, 400,000, depending on really who you ask. And of course they have their own family tree, if you like. They have their own phylogeny. You can see this is related to that and so on. And all of that, well, quite a lot of it was sorted out by having a look at the characteristics of the plants, the leaves and the stems and all of that sort of thing, just like with all of the other plants. But the base of that tree, right down in the early diverging plants, wasn't really terribly well sorted out. And there was quite a lot of debate about what was this earliest diverging plant. They thought it might be something like a magnolia, which you can see outside, or maybe one of the Austrobaileales, which are over there in the corner of the glasshouse. And then phylogenetic research involving lots of DNA studies rather unexpectedly revealed that it was probably this rather unprepossessing looking shrub that we have here. This is Ambarella trichopoda. We're amazingly fortunate to have a specimen of it here on display in the Botanic Garden. It's very, very rare to find it in a Botanic Garden, and the only other place you can see it in Europe is in Bonn. So, like I say, really, really great to have it here. It's got these rather odd laurel-like leaves. They're quite leathery and hard. It grows in New Caledonia, which is an island about 700 miles off the east coast of Australia. And it does have flowers. We've got some little tiny flower buds, which we're hoping will develop into flowers fairly soon. There's no real differentiation between petals and sepals. The plant produces either male flowers or female flowers in one year, but it can switch between years, so it's not the plant that is male or female, but the flowers. So here we have it, one of the examples of the earliest diverging flowering plants.
We're standing here next to Magnolia, which is one of the earliest derived angiosperm groups. So the Magnolia plants have very simple flowers. We can see here it's a very large, white, open flower, very simply shaped like a cup or a saucer, and the pollinators, which are usually beetles, access the pollen and nectar reward in the middle without having to deal with any complicated floral structures. It's a nice, simple flower. But as the angiosperms evolve, the flowers do become more complicated, and we'll show you that in some other examples as we walk round. We're going to talk about the monocots now. The monocots are one of the biggest groups of the angiosperms. About 25% of the species diversity of the flowering plants is in the monocot group. And the monocots are very distinctive. They have usually long strap-shaped leaves that have parallel veins in them, so they can look very different from the leaves and the venation patterns that we see in other angiosperms. And they also have very noticeably distinct flowers. The flowers have their parts in threes, so normally 3 petals, 3 sepals, 3 stamens, and so on. And those flowers are very distinctive because those 3-whorled structures are specific to the monocots, and they produce flowers that you'll instantly recognise, like lilies and irises and daffodils and so on. When people think about the monocots, they very often think not about the palm that I'm standing next to here, this is Trachycarpus, but about the Poaceae family, the grass family over here. And the grass family is, of course, the most economically important family in the monocots, and indeed one of the most economically important families of plants. All of our cereal crops, wheat, rice, maize, and so on, come from the grass family. And so it's easy to think, when you think of monocots, just of grasses. And of course the grasses are monocots, but there are lots of other monocots that aren't grasses. Not just this wonderful palm, the lilies and daffodils and so on that I mentioned, but also, perhaps most spectacularly of all, the orchid family, one of the most species-rich families of plants there are, with those amazing floral structures, is also in the monocots.
The other main group of angiosperms is the eudicots, and within there you'll find 2 main groups, the asterids and the rosids, and I'm going to talk to you about the rosids. Within the rosids there are again 2 main groups, the fabids and the malvids. Now the fabids contains all sorts of commercially important crop plants. You've got things like the legumes, peas, beans, soy, that sort of stuff. You've also got the rosales, which contain a lot of commercially important fruit crops. Strawberries, raspberries, apple trees, pear trees, cherries, almonds, they're all in there. Also, we've got the cucurbitales, which contain pumpkin, melon, squash. Again, lots of really useful crop plants in there. And within the fabids, you'll also find a nitrogen-fixing clade. Now, they form symbiotic relationships with root bacteria, rhizobia, which fix nitrogen from the air and provide the plants with these nutrients which they can't otherwise get from the soil. So it makes them able to grow in soils which they wouldn't otherwise be able to grow in. Now the reason I'm sat in this bed is because the other group is the malvids, and I'm sat next to Althea officialis, the marshmallow. It's a bit different from the marshmallow sweets that you get. You can make those marshmallow sweets with this, but they're really not very nice indeed. And also next to me here we have Hibiscus trionum, and this is one of Beverly Glover's model organisms for use in research. It's got an iridescent centre to the flower, which shimmers in the blue range, which means that it's easier for bees to see. Also in the malvids we find the brassicales, and brassicas again are really important crop plants of cabbages and oilseed rape and things like that. But most plant scientists will probably know the brassicales best for the workhorse of the lab, which is Arabidopsis. That's in the Brassicaceae. That's probably the most widely experimented on plant around the world.
The final plant group we want to tell you about is the asterids. That's the other major clade of the eudicots. There are about 75,000 species in this group. It's a really important group of flowering plants, and it includes lots of commercially important families. Within the asterids we have the Solanaceae. That's the family that includes tobacco, tomato, potato, lots of important crop plants there, but also very useful model plants for plant science research. There's also the Lamiaceae, a family that includes plants like snapdragon that you might grow in your garden. And perhaps most species rich of all, within the asterids we have the daisy family, the Asteraceae, like these beautiful daisies I'm sitting with here. This is a very species rich group with a real diversity of flower structure and colour, all very different from one another, but all based on the idea of having multiple floral units combined in a single structure that we call a capitulum that looks like a single flower. That's the daisy itself. So the asterids are a really big group, really important, and contain quite a lot of the species diversity of the plant kingdom. Well, that's the end of our trail. Thank you for joining us on it. In the last half hour or so, we've taken you on a tour through about 450 million years of plant evolution, all the way from Chara, that representative of the green algae groups, through to the flowering plants, including some of my favourites like this wonderful clock vine here, Thunbergia, with its gorgeous bird-pollinated flowers. So I'd really recommend, if you get the opportunity, to come to the garden and do the trail for yourself. There's nothing to compare with seeing the plants in real life, in touching them and in smelling them. So if you do come, you want to ask for the trail leaflet at the ticket office. This is the Evolution of Plants trail. They're free to collect at the ticket offices, or you can download it before you come and use it that way instead. So please do come and have a look at the garden if you possibly can. You'll get far more enjoyment from that than just from watching us, but we've enjoyed spending this half hour with you, so from Hamish and me, thank you very much and goodbye.